Okay, why don't we just go ahead and get started? I'll do a brief little introduction and then we can start at 11:10. My name is Todd Hagobian. I am the co-director along with Suzanne Phelan. She's the director of the Center for Health Research. And I'm a professor in the Department of Kinesiology and Public Health. And we want to welcome you to CHR's COVID-19 Research Showcase with our Cal Poly community. We had an unbelievable amount of submissions. And those that are presenting today are representing work done from every college on campus. And then we hope you'll visit our conference folder. And I put that link in the chat already to review all the full list of abstracts selected for the presentation. And you'll see a copy of the slides with the schedule for both tracks. And so thank you for attending and participating in the Virus and Impact track. And before we begin, a few words about the Center for Health Research, or CHR. Our mission is to facilitate transdisciplinary research in the prevention and treatment of chronic diseases, which are the leading cause of death and disability in the US. Our center's core values include promoting health equity and improved quality of life across the lifespan, and that minoritized populations with obesity and chronic disease are more likely than other people to be affected and to die from COVID-19. So we felt this was a natural fit for CHR to host this showcase and just the amazing work done at Cal Poly and to understand and combat this pandemic. And if you aren't yet involved in CHR, I'd like to encourage you to take a moment to visit our website, reach out to the CHR rep. So we have a rep in each college, it's a faculty member, and sign up to be an affiliated faculty member or student. And I will put that link here. I have that in the chat.
I got you covered, Todd. I'll do it. Okay,
thank you. Thank you, Suzanne. And then I also want to put a link here. This last link, I have all these links here, is to the website for this symposium that we have on the CHR site. And then eventually we will have the recordings posted on there, and all the abstracts will be posted on there too. And so just as a brief overview, this is going to be their standard slideshow. The presenters have up to 10 minutes to present, 5 minutes of question and answers. If you have questions, can you please hold them to the end? You can put those in the chat, or you can raise your hand if you would like, and you can ask those verbally, either way. And then we'll move on to the next talk. So our first talk today is, and the title's Identifying Inhibitors of SARS-CoV-2 with a Virtual Screen. And I will have the authors introduce themselves.
Hello, everybody. My name is Dr. Scott Egan. I'm an associate professor of drug discovery and medicinal chemistry in the Department of Chemistry and Biochemistry.
And I'm McLean Howland. I'm a computer science student, and I'll go ahead and get started with the presentation. So our research was working on identifying inhibitors of the SARS-CoV-2 virus using virtual screening techniques. So as you all know, it's a big pandemic. We've got 155 million infections, nearly 3.2 million fatalities, and here in the US, we account for nearly 18% of those deaths. And we've got more than 1,000 detected strains, with new ones emerging all the time as the virus continues. So we were looking for ways that we could help with this. So the way we did that was by targeting a viral protease, or a viral protein called Mpro. Now, a protease is a protein used in the replication cycle of the COVID virus, and it's a protease, so it's responsible for chopping up long strands of other viral proteins, like a big pair of molecular scissors. And if you take a look at the Mpro complex on the right, you can kind of see its quaternary structure. You've got, well, a big thing to point out there is the purple residues. Those are kind of like the tips of the big molecular scissors, and those go and cut the other viral proteins. And you can see in that figure, we've put a small weak inhibitor in there, the green molecule that is stuffed right in between those purple residues and will hopefully inhibit it. Another important point is that humans don't have any analog to Mpro. So if we find a molecule that inhibits it, hopefully it won't have major toxicity issues with humans. And we also started working on this research before a vaccine was released. And it's important to kind of have small molecule inhibitors or drugs to treat an infection of the virus, because people may not be able to get the virus or may not be able to get the vaccine, or they may already be infected with the virus. So it's important to kind of have both in your arsenal of treating this. The way these small molecule inhibitors work is by kind of fitting themselves into the active site of the protein right there, like with the green molecule in this diagram, and by stopping it from working. So it's kind of like stuffing a key into a lock and then breaking off the key, and where the lock is your enzyme or protein and the key is your small molecule inhibitor. So the way we went about finding a small molecule inhibitor for this protein was by using virtual screening techniques. So there's lots of databases on the internet containing millions of compounds. We chose one for this screen that contained about 100 million compounds that are commercially available, meaning that theoretically we could go and order them and have them show up to our door. So we took those 100 million compounds in the Zinc 15 database, and we applied some filters to get more drug-like compounds that look like existing drugs we have already, just so we can pare down that 100 million compounds into a more manageable 7 to 13 million compounds. And for this screen, I think we ended up with about 9 million compounds. So then we take those 9 million compounds and we run them through a computer simulation to estimate how well they bind to that active site of Mpro. And we get a big ranking, and then we take our top inhibitors and do some more tests with them. So now I'll talk a little bit about how we actually rank that binding energy. So we use something called a docking program to estimate how well the small molecule inhibitor or ligand fits into our target protein, which is Mpro. So it takes the small molecule and runs a bunch of simulations and tries to optimize how it fits in that active site of the protein, and spits out a number that represents the binding energy. And in this case, a more negative binding energy means a better fit because it releases more energy and is more stable. And we used a program called iDoc for this screen. Now, one problem that you run into with these virtual screens is that we've got 9 million compounds and we want to screen them all. If we were to screen them one by one, each one takes about 5 seconds. So 9 million times 5 seconds results in about 1.5 years of computational time. But obviously that's a little bit too long, so we had to find a faster way. The way we do that is by using distributed computing. So we've got big computer labs on campus. The one we use for this screen is called the MPAC lab. So we've got 30 high performance computers all connected on a network. And so I wrote a program that goes and distributes the 9 million compounds over those 30 computers and runs them all in parallel. So we've got one master computer that distributes the ligand files and tells the workers what to do. The workers run those docking simulations and then send the results back to the master. And once we're done running them, we compile the list of all 9 million compounds and sort them by the binding energy and take the top couple ones. I know I saw a couple computer science faculty and students in here. So if you're curious, I wrote it all in Python and just running on the MPAC labs using OpenMPI. Now, the results of this virtual screen. So we took our big list of 9 million compounds ranked by binding energy, and we took the top 75 compounds. And then we subjected those to more filters. So we filtered out the top 75 compounds for things like toxicity, or things like functional groups that are almost guaranteeing toxicity in humans. And we also filtered out really similar compounds so that we weren't wasting effort testing, running more tests on really similar compounds so we can kind of have a more diverse field. So that resulted in 28 compounds out of our 75. And you can see some of those examples on the right there. We've got the Zinc database ID, you've got their energy score and their rank and structure. So once we had these 28 compounds, we went and tried to order them. So ordering them online, we got 20 of the 28 compounds on our door. So we packed those up and sent them off to Germany, the University of Lübeck, for more bioassay tests. And we only were able to get 20 of the 28 compounds because even though it's listed in a database that says it's commercially available, it's not always. You might not be able to reach the vendor. Sometimes they're just unavailable. So after we sent our compounds off to Germany, our collaborators over there ran a fluorescence resonance energy transfer based cleavage assay, which tests the inhibition of the small molecules we sent them against the cleavage activity of Mpro. And so in this case, 100% inhibition would be Mpro didn't work at all with our small molecule inhibitor, and 0% would be Mpro worked completely fine, even with the inhibitor. So we identified two compounds, CP13 and CP25, that showed 45% and 35% inhibition of Mpro. You can see those on the upper right-hand corner with their structures. Then we took those two and subjected them to more intense computer simulations, molecular dynamic simulations using the AMBER-18 program to kind of further estimate how stable they would be when they bind to Mpro. And you can see that on the bottom right. You see the Mpro complex in white behind it and the inhibitor CP13 in orange right there. One thing to point out is if you see those purple residues that are the tips of the molecular scissors, it's kind of stuffing itself right near them and hopefully stopping them from working. So we identified two kind of top hit compounds, CP13 and CP25, but the 45% and 35% inhibition aren't necessarily potent enough to advance into clinical trials. However, these things do serve as good starting points for optimizing and creating more powerful compounds. One way you can do this is by attaching electrophilic warheads, just more functional groups to the molecule to make them more potent. And kind of a fun fact about this is that Pfizer actually recently started early clinical trials for a compound that targets the exact same spot that we were targeting in Mpro. And they found a compound that was potent enough to advance to the clinic. So it is pretty cool to see that this line of research could result in an actual drug. And you can see the structure of the Pfizer compound down below there. And notice they've added an electrophilic warhead, kind of like we were thinking about. So that it was a pretty cool experience. And this project was funded by the Bill and Linda Frost Fund at California Polytechnic State University and the European Commission through the SCORE program. Thank you. Any questions?
Thank you, McLean. That was great. So this paper is now open for discussion or comments.
Let's see. Amazing. Will you work to increase potency or move on as Pfizer is on it? We'll probably move on as Pfizer is on it. They've got about 200 PhDs that came up with this and will continue to work on it. So I think we'll leave that to them.
Let's see. Are there other questions? I have to see if I can, if anyone put their hand up. McLean, I have a question that this is great work. And you, for the Mpro, you mentioned that the two compounds had 45 and 35% inhibition, and that wasn't enough. What is, like, how much inhibition is good enough? What would that be?
I'm going to pass that off to Dr. Egan, who's the medicinal chemist.
Because it seemed good, Scott. Is that good enough, or is that not? It needs to be better.
It's not. It wasn't bad, but to get those numbers, they had to be at pretty high concentrations. So theoretically, you could have driven it much higher. You start getting into large amounts of drugs. So at the level that they were testing at, we'd really want something to be 80% or better, and that's where Pfizer's compounds are. They're up in the high 90s. So we gave it a good try, but we weren't successful. But anytime you do a project like this, you're always a part of a big team. Someone somewhere will find it. So we were just glad to contribute. But yeah, 80% is kind of the threshold there.
Any other questions or comments?
I just have to say out loud that I'm just so impressed at all of the everything that you did is incredible in such a short period of time, and just your presentation skills, everything is just very impressive work. So congratulations on that.
Yeah, thank you.
Great, thank you. So we'll conclude our first presentation. Yay. So we'll wait till 11:25 and we'll start our next talk. So a few minutes here, few minute break for our next talk.
Oh, I thought we were short on time, so I could have asked just another quick question.
No, we're still on. We have.
Yeah, I was
guys,
like,
go
I didn't
ahead.
want to know what time, how much time we had. But what were they doing in Germany that we couldn't do here? Had you thought about that? What? Just talk a little bit more, if you can, about what their role was and what they did.
So I guess a big omission on our part is that our collaborators in Germany are the people who originally sequenced and figured out the structure of the Mpro protein, right? So they had copies of the protein and the ability to make it and do tests with it. So we sent off the compounds to them to do that. I think we were kind of lucky because they came out with that, they published it, and everyone went wild, and we managed to slot in and get them to help us out.
I see. That's great.
And there are also biosafety hazard concerns with having a virus like that on campus. So, and frankly, I don't like working with those viruses, so I'd much rather having someone with expertise on that. But you know, they were the ones that discovered it, so we didn't think we could do better than them. So we worked with them.
Makes sense. Thank you for that.
Okay, we'll start on our next talk in just a minute here. I just wanted to, if you didn't notice in the chat, I will re-put the 2 SharePoints here that has the information on all today's talks, including the schedule and about CHR. So that is in the chat. And then our next talk is titled "A Simple Fix for SIR and SEIR Pandemic Models Tested with Particle Simulations" by Dr. Fernsler.
Yeah, thanks very much, Todd. So yeah, my name is Jonathan Fernsler. I'm a physics professor here at Cal Poly. And I became interested in epidemiological modeling a year ago, like many people did, because of the impacts of coronavirus. And so today I'm going to talk with you about a student project I've been working on for the past 5 months, where we are using particle simulations of epidemics as a way of testing out some of the dominant models that are used today by the groups that contribute to the CDC forecast. And those students, I just want to go and name them because, and I see some of them are in the audience here. I've got Julianne, Peter Lentz, Alexis Morris-Solik, Madison Singleton, and Patrick Sullivan. I also want to call attention to my father, Richard Fernsler, who this is the first project we've ever been able to work on together. He is a physicist as well, and he worked on with me for the last year on the theoretical background to the project. So it's been a really neat experience. So what we have a short time, and we actually have a large number of conclusions we've reached from this project, and some of them we actually feel are provocative and surprising, and we hope this will trigger some conversations with the epidemic modeling community. And so I'm going to jump right into our main conclusions to start my talk, and then I'll go and take a step back with the time we have left to go and do a little more background. So what we did was to simulate epidemics using particles, where each particle represents a person. And we can classify those people's relationship to the disease as people who are susceptible to the disease. And he'll run our little simulation here. And if a susceptible person comes nearby an infected person in red, then there's a probability chance that they may become infected. This is determined by a random number generator to see if that propagates. Those people then become infected for a certain amount of time that we can set in our simulation, and then they become recovered and are no longer infectious. So a very important other aspect of modeling these diseases that we found is that people's infectiousness changes as a function of the time after they've been infected. And so we can represent something like this as a probability of infectiousness type function, which is again, after the time that they've been infected. And this type of function has been measured for COVID-19 using contact tracing data. And we can generate a simulation then that reproduces this type of behavior. And so this is kind of like an actual modeled coronavirus mini epidemic. So what we wanted to do with these particle simulations was to compare some of the main models that are being used today to predict the coronavirus infections used by the CDC. And the dominant model that I've found from surveying the CDC website is the SEIR or SIR models. And these two models are very common and are used by many groups. We wanted to compare this to a second model that I'm coining as the general infectiousness model. And this model is much less well known, although it turns out that its historical roots are just as old and possibly older than the SEIR and SIR models. And so we believe this is a first comparison that's been made of actual solutions to these models in the literature. And so we're hoping to finish writing this up and publish this hopefully in the near future. So the first thing that an epidemic model should be able to do is to predict the infection rate. And this is similar to the daily new cases you see reported from Johns Hopkins. So what we did with our project was we used the early data to our simulations, our particle simulations, and then fed them into these two different models to see if they could predict what happened in the future. And we used all the standard tools that are used in the literature by the main CDC groups. So our particle simulation here is shown in red, and the SEIR model would do a reasonable job with some of the predictions, but when a change was simulated, then they often did much more poorly, like this one where we're simulating wearing of masks. On the other hand, the general infectiousness type of model was able to adapt to those changes and generally did a better job simulating or predicting the infection rate from the simulations. The other thing that an epidemic model should be able to do is to predict the fractions of the population who are in these susceptible, infected, recovered type states. And so again, we can do this with our particle simulation and try predicting it with the different models. And again, we found that in all the cases that we have investigated where we expect these disease models to be able to work, the general infectiousness model did a much better job at predicting the data than the SEIR model, which is surprising because it is much less well known. So we believe that there are some significant flaws to the SEIR models that are being used. And we believe that there is a simple fix because the general infectiousness type of model is actually just as easy to implement, and our students wrote the code for all of these. So these are absolutely applicable types of models that can be used. Okay, so with the time I have left, I'd like to go and give a little background to epidemic models. So epidemic models are mathematical tools that are helpful for governments to plan the future impacts of an epidemic like coronavirus. And here I'm showing the IHME lab model for California. This is back in December. And they predicted a peak in infections over the winter months. Of course, we indeed saw that occur in California, and it had huge impacts on the state and around the nation. So they are using an SEIR type of model, as I described. When I've surveyed the CDC website, they use many different models, 29 in total. And the dominant model by far, there are a variety of models, but the dominant model is this SEIR type rate model. And as an example of that, 6 out of the 7 models are used by the CDC that use social distancing changes of people, basically changing people's behavior to predict the future outcomes, are SEIR type of models. So these models, just in a very brief description here, can be, they automatically classify the population into those categories of susceptible, infected, and recovered. And they use the number of infected people to predict the future infections. They do this by using these parameters, rate type parameters, that are related to the famous reproduction number, R0, that I think probably everyone has heard of in the media at some point. So these rate parameters are similar to, can be thought of as like little knobs you can turn to go and adjust how people's behavior could change and then predict how the model will adapt to it, like people wearing masks or social distancing or quarantining, etc. These parameters must be interpreted from the infectiousness probability function, and they use fits to the infection rate data to seed the model. So our students were split up into 2 groups, where we had one group of a particle group that ran the simulations in a variety of different circumstances. And you can do all sorts of different things with these simulations that can try to mimic the behavior of real people. The second group, the modeling group, then took that initial data from the simulation, which is comparable to the daily new cases you see here from San Luis Obispo County. And here's some example data right here. So for the first 10 days of a simulated epidemic. And then they use that to try to go and predict, using standard procedures in the literature, the next 90 days, say, of the epidemic. Okay, so I'll leave with one last little case study to show again what we're talking about. And for this one, we looked at the impact of population density on the spread of an epidemic. So if we compare early data from the coronavirus epidemic back in February and March, New York had a very rapid rise in cases in comparison to a state like Wisconsin that had a much slower growth initially. And a lot of this can be attributed to the difference in population density between these states. Later on, as an epidemic progresses, then other behaviors can be more important. But initially, a lot of this is attributed to things like population density. So we can go and simulate this with a particle simulation by just reducing the density of the simulation. And we think that a good epidemic model should be able to predict the differences. So if we run this with our SIR model, then we found that it had a reasonable simulation or a prediction of the urban situation. But if we try to go and use those parameters to figure out what we think would happen in a rural simulation, then it was much more poor. Similarly, it undercounts the peak number of infected people in all of our simulations. So what we found from this is that the SIR model is flawed because the parameters that seem to be simple rate parameters are actually more complex. They are not independent of one another. So if we change the reproduction number, then in order to predict the future of the epidemic, one must also change the recovery rate, even though the particles in our simulation were infected for the same amount of time in both simulations. Okay, so it's a more complex kind of interaction between these parameters than it first seems, and this we believe could lead to a lot of errors being made. So in closing, our general infectiousness model we believe is a fix for these flaws, and this model uses that probability of infectiousness function directly to the infection rate data to predict the future, and we found that it worked better than the SEIR and SIR models did in all the cases that we have investigated. And if we go and compare those results, again, we can see for our population density simulations it does a significantly better job, and it adapted to those changes. Notice, by the way, that the number of infections here is actually much lower for the rural simulation than the urban one. And again, also for our population fractions. So in conclusion, I'd just like to say that it's been an interesting project, and we believe we found some serious problems with these dominant models, and we'd love to engage in more of a conversation with people in the epidemiological community about these things we found. Everyone is inundated right now with different models and ideas, but we'd love to go and talk more about these findings. And also just want to thank my students. They did an incredible amount of work in 5 months' time. They learned a ton about modeling and programming and other issues. So thank you very much. I appreciate any questions.
Thank you very much. That was a great work by you and your students. And so we have a few minutes here. We have time for some questions or comments.
One thing I've wondered about with a lot of these models is to what extent they incorporate heterogeneity in the population. Do your particles have different collision rates, or can that be built in?
Great question, Jason. Yeah. So all of these, the models that I've been investigating assume a homogeneous population. Okay, and those are the ones that are actually used by these CDC groups. Now, the way they tend to do it is they'll break up the population into usually the county-based, just as Johns Hopkins does. It breaks up into county-based reports, and basically the assumption is that within a county that it's a relatively homogeneous population. But you're absolutely right. What's something we would like to investigate in the future. We haven't been focusing on that as much yet, but we'd like to go and investigate what happens with our particle simulations if, we have it where it's more heterogeneous, it's not homogeneous. And so this, we can easily do things like this by slowing down the particles, for instance, and you just have little pockets of infection that will go and spread in one region, or you could have more complex interactions as well. So that's something for the future for sure.
Great, we're almost out of time here, but Dr. Fernsler, I just want to add a quick question. Can you use this model? You said for infectious disease, but can you use it for other diseases like chronic diseases? Maybe the social exposure, would the same principles apply for that? Yeah.
What's really neat about these models is that they are applicable to a really wide variety of circumstances. And for instance, like the sort of seminal paper that brought mathematical modeling, epidemiology into the biology community is a paper in 1981 by Anderson and May, and they actually use this for the study of microparasites in insects. And that's then became the seed to use it for diseases in humans. So there's a wide variety. These things are can be thought of in a mathematical kind of way, and so they have wide applicability.
Great, thank you for your presentation. We didn't move on to our next talk, but Dr. Fernsler, there is a question in the chat. Maybe you can look at that, and if you can answer that, that would be great. And so thank you all for attending. This has been just a great presentation thus far. And our next up is the modeling the spread of the COVID-19 over varied contact networks by Dr. Migler, and I'll want you to introduce yourself and your student.
Hello, I'm Dr. Migler, and I'm an assistant professor in the Computer Science and Software Engineering Department. And my student, Ryan Solorzano, has been doing excellent work for his master's thesis. And it's a nice follow-up to Dr. Fernsler's presentation because we're using contact networks, and Ryan is slightly modified the SIR model. And he's going to talk about the impact of just testing and quarantining, and what testing and quarantining, what sort of outcomes we expect with that. So Ryan, please go ahead.
Sure. Let me go ahead and share my screen then. Okay, and I'm assuming that should be visible, so just let me know otherwise. So So yeah, my name is Ryan Solorzano. I'm a master's student in computer science, and we'll be talking about modeling the spread of COVID-19 over varied contact networks. So like we said, we focus more on testing rather than simulation and just kind of seeing which testing strategies are the most effective. So a quick overview of contact networks. Contact networks, the vertices can represent organisms, and two organisms can be connected if they come into contact in a way to allow spreading of the disease. So this can be dolphins or, of course, people. So in the case of our research with COVID-19, the vertices are, of course, people, and two people can stay connected if they breathe that same air to allow transmission of COVID-19. So here's an example contact network. We can say like maybe A, B, and C are all roommates, and so that's why they're all interconnected. And then maybe E has a class with C, and that's why they're only connected with C and not with B and A. And then let's say maybe D is a club sport with B and C, but not A. And then we can also introduce this idea of a temporal graph, where basically maybe at night, A, B, and C are connected, but E and D are not. And then again, like maybe during the day, during club sport time, B, C, and D are connected, but not A and E, and so on. So this is kind of the overall idea of contact networks. So the collaboration we used was from a collaboration called SocioPattern. So SocioPatterns is a collaboration between researchers and developers around the world to collect this contact network data and build these networks. And so we use 5 different contact networks from these SocioPatterns. 2 are actually office workplaces, 2 collected 2 years apart, and 1 is collected from a high school, another is from an elementary school, and another is from an academic conference. And I'll get into a little bit of the validity of this, but then in all 5 contact networks, 2 people are connected if they come into 1.5 meters of each other, or about 5 feet for a 20-second period, and they all use the same infrastructure to collect this data. And we could accept this to be a reasonable approximation, breathing the same air for 5 feet. And we use, and this list content networks collected using RFID tags. So if anyone's it's done like a 5K with like a racing number, they have like a chip on the back to collect when you finish, that's an RFID tag. And so it's the same technology. And let's talk a little bit about the model we use. So we have this SEIRQ model where each person in the network can be in one of 5 possible states. And that's susceptible, exposed, infectious, removed, and quarantined. You notice I changed it from recovered to removed, and that's just because it encompasses all possible outcomes of the disease. So basically, everyone starts out in their model. Every vertice starts out with susceptible, and then to start the outbreak, we move 1 to infectious. This can vary based on simulations, but in ours, we chose to use 1, and we changed the person to be infected every time. And then in the content network, we basically just play out the data. And if someone in infectious comes in contact with someone susceptible, you have a certain amount of chance. For us, we use 3%, which we kind of tweaked based off of research we had. But basically, there's 3% chance to be moved into this exposed bucket, which basically guarantees that you will eventually contract the disease. And again, infectious after a while, let's say it varied again on research, but somewhere between a week or 2 you can be infectious and then eventually be moved to removed, and then again exposed, you're exposed for a little bit of time, and you have some sort of chance of you eventually move to infectious, and at any point we also have our testing strategies. So basically our testing strategies test anyone in this network, and you have some chance of testing positive based off if you're exposed to infectious, again, based off of research on these tests, and then you can move to quarantine. And an important part of this is that we assume a perfect quarantine, meaning that they're completely removed from the simulation, so like no contacts can possibly happen. And after the 2-week period, you can be removed. And then we also include the possibility of a false positive test, moving from susceptible to quarantined, and then after that 2-week period, moving back to susceptible. So talking a bit about our testing strategies, we have the easy one where you don't test anyone, basically just seeing what happens with our outbreak. And this can be considered our control. And then we have random batch testing. So this is also a bit of a naive approach where you basically just get a subset of the population, the size of which depends on the number of tests we have available for that day, and then just test them and quarantine them appropriately. And then we have individual testing. So this is a bit inspired by what we're doing at Cal Poly. We're basically, again, developing an availability test. Every single person is regularly tested. So let's say a person can be tested every 3 days, for example. And again, the amount that person is tested is depending on availability of tests. And then we have testing using degrees. So this is a little bit more graph approach or graph theory approach, where basically degree of a person is how many people they've come into contact with the previous day. And so the higher degree you have, the more likely it is to be a super spreader, because you, one, have a lot more chances to be contract the disease, but also two, the more important part is that you have a lot of chances to spread that disease. And so the idea is, hopefully, if we identify these people early on, we can mitigate the spread a lot quicker. And let's get a little bit to our results. So this is results from our high school network. Again, so to be clear, the vertices represent students and teachers, and the edges are just whenever they interact. And this is a content network of about 326 individuals. And again, we assume perfect quarantine, so that would just mean that they don't go to school for 2 weeks. And then again, this is collected over a 2-week period. And but our simulations ran for several weeks. And so whenever we ran out of data, we just loop the data, which is a very reasonable assumption to make, especially in a high school setting where people are creatures of habit, people have the same schedules, meet with the same people, same friend group, so on. And so that's what we do here. And let's look a little bit about one more note. We also don't assume any sort of behavior change, such as mask wearing or social distancing or making pods within classrooms. And that's just because we want to see just purely just the tests. And so again, this graph on the x-axis, we have the number of tests per day going from 0 all the way to the entire population per day. And then we have on the y-axis the maximum number of people infected in our simulation. So if the infectious bucket had like a graph of number of people, it is eventually going to peak at some point and then go down. And so the peak is what we plot here. And each plot point here represents 10 simulations averaged out. And so that we have like a very clear outcome. And so this red dot here represents no tests. It's about 160 people peak infection. And then strategy 1, the random batch testing, that follows this nice little curve all the way down. And strategy 2, the random individual testing, interesting enough, is kind of hard to distinguish from the first one, where basically there's not too much of a difference if you just get a random batch of the population versus if you get if you guarantee that every single person is tested. And so that's very interesting. And then strategy 3, if you look at this, it seems to do a little bit better until about halfway, where it just kind of becomes random noise of who becomes better. But generally with first like half, it seems to perform better than the first two. And this is actually very interesting and actually very good that we see this, and we're happy with these results. And then here's what this looks like at an office workplace. Very similar, just a little bit more sparse because the network is smaller. And then at an academic conference. And this academic conference was collected over a period of 2 days. And so maybe not necessarily realistic to loop those over for weeks and weeks. However, it's important to note that academic conference had a lot more dense of a network. And so we can see that the degree became a lot more important early on than the other strategies. And so that's just reflected by the density of the network. So some future work really quick. So basically any future work can improve the realism of the simulations and strategies. So one point is the asymptomatic and symptomatic buckets. We have proved that asymptomatic people can still spread the disease. However, we didn't necessarily take that into account. And also you could just test symptomatic people and so on. You can also look at contact networks where strength of the signal is indicated. So you can kind of vary that infection probability. Strategy with perfect contact tracing would be interesting. And then a combined graph theory strategy. And then something I didn't mention here that I didn't know about is something we saw with a particle simulations with the different just different simulations of like altogether. And then also simulations involving 2 doses of vaccine with varying efficacy could be interesting, so that you could target the right population and stop the spread of the disease. And then combining content networks to make a community would be very interesting, such as like, let's say you have a workplace and a preschool and you make like families within that unit, and just to make it more realistic and more granular, to make like a very like realistic community could be interesting. So here's some of our references really quick, and then that would be a good time for questions.
Thank you, Ryan. Wow, that was a great presentation. And what you made, just of note, those graphs and figures are really well designed. And we know there's a lot of data points in there, and it took a lot of work to do that. So we have a few minutes for some questions or comments. Dr. Fernsler, yes.
I have a question. Yeah, so yeah, really great work. And I love that, because this is like on the other end, like exploring sort of the heterogeneity, you know, kind of diseases, which is the opposite thing of what I'm doing, which I'm really, it's really cool. So just I was wondering, is there a simple prediction for you know, your outcomes based on like the network density? I mean, can you sort of, is there a simple model that describes like, you know, how testing would behave? Because you said like the faculty network, you simulated one network density, and then the high school a different one.
Yeah, that's actually a really interesting question. And like you saw, like that curve was seemed pretty smooth. And so we could actually look into that while we're still like collaborating all of our research. And so this is actually like that research, or the results were very recent. And so that's actually very interesting to look at, but it seems possible, right? Like it seems like there was a distinctly the more dense networks did better. And so that's something we can look into.
Cool, thanks. Yeah, great work.
Thank you.
And one thing to note is that the networks that Ryan the networks he was using were actually they were much denser than you would expect a real life network to be, because as he alluded to in the future work section, these are small, super interconnected networks within the larger network. And so there was a lot of density that if you considered a whole, you know, community, a whole city, the density would go way, way down. So,
Great. We have time for one other quick comment or question before we move on to the next talk. Any comments or questions? Well, thank you very much. What a great presentation.
Thank you.
And so thank you for those of you that are just joining us. And I wanted to put in the chat the schedule for today and some slides on the Center for Health Research, so you can take a look at those. And then the recordings will be posted online along with all the abstracts. And so we're gonna move on to our next talk. And so the next talk is titled Inventory Levels and Food Prices During Peak Food at-Home Demand Periods, Assessing Pandemic Shocks in the US Food System. And I will like Dr. Volpe to introduce himself and his students.
Hi, everyone. Hey, so thank you very much for joining today. I'm really excited to showcase some of the work that my colleague, Dr. Xiaowei Tsai, and myself have been working on for the last year or so. It's funded from the USDA Economic Research Service, and it's kind of a long run project trying to measure when the pandemic hit, what happened to the food supply chain, what happened to food access and food prices and food purchases, with the long-term goal of trying to have a food supply chain that's kind of more resilient the next time we get hit hard. So with that, I really want to turn things over to the students. This is their show. So thank you all very much for tuning in. I appreciate it.
Awesome. Good morning, everyone. Like Dr. Volpe said, thank you for having us. Let me just get this, share my screen with everyone. Awesome. So today we will be presenting on inventory levels, shopping baskets, food prices during peak food at home demand periods. Like Dr. Volpe said, this is a cooperative agreement funded by the USDA Economic Research Service. And this presentation was made by and with the help of Dr. Richard Volpe, Dr. Xiaowei Tsai, Maxine McFessel, myself, Camille Hardiker, and Katie Denise. Just a quick disclaimer that the view in this presentation are those of the authors and do not reflect the USDA or the ERS. So the long-term project objectives are to identify retail food stockouts and shortages and develop a data set of their locations and magnitudes, as well as to identify price impacts of the pandemic and related food shortages. Lastly, another goal is to measure potential food access impacts of the pandemic. So this is all necessary because traditional food scanner data does not provide information on food stockouts or shortages. Therefore, the research team presenting today are engaged in efforts that will help identify such stockouts and shortages. So I will be presenting today on behalf of the database team, which consists of myself and Rona Haworth. So some of our objectives and main tasks were to find and analyze data to aid in measuring and identifying food shortages due to COVID, changes in transportation capacity, specifically barges and trucks, as well as wage data on a county level, and how other factors might affect food shortages other than COVID-19. So we used a variety of publicly available information from numerous sources. Most of the federal data sets we use are from the USDA and the BLS. And the following slides all show examples of insights related to the pandemic that we have drawn from this publicly available data. So when restaurants and bars closed, food spending in the US shifted dramatically towards supermarkets, which contributed to shortages and stockouts in the early months of the pandemic. Compared to 1 year prior, many food prices increased sharply during the onset of the pandemic. Meat production was particularly problematic and large quantities of fruits and vegetables were wasted. And this led to particularly high food prices for these specific food groups. There were many costs affecting the food supply chain, which rose during the pandemic, but by varying degrees. So once again, we see that animal processing and meat production were hit very hard early on in the pandemic. Food at home and grocery prices saw their biggest 1 month increase in over 30 years in April of 2020. And then this slide and the next touch on the transportation industry, the trucking industry in particular, which faced significant problems even before the pandemic, mainly due to driver shortages. These slides show that since COVID-19 began, truck rates have been trending up while truck availability has been trending down. This has certainly contributed to food price increases and food shortages in the last year. So some of the key findings Rona and I found were numerous data sets found describing various commodity availability, transportation availability, producer price index for various commodities and changes by month, data specific to COVID-19, which was awesome, weekly advertised fruit and vegetable retail prices by region, including the number of stores that stock the commodity, the units sold, and the variety. And there is currently acquisition underway in collaboration with the USDA and the ERS for some of the data sets listed in the previous slides. Next, I would love to introduce the news team, and Camille Hardiker will be speaking on behalf of them.
Hello, my name is Camille Hardiker, and I work alongside Joe Shea and Haley Powell on the news team. We help identify where shortages happen through popular press postings in effort to help the entire team find the food shortage data not recorded by scanner data. We collected public information to understand what food and beverage products and regions saw the most shortages during the pandemic, as well as what the main reasons were for these shortages. Our data was pulled through Google searches using key search terms, social media posts, and online videos before being sorted and analyzed. Here we have a list of resources that we have used throughout our research process. These include digital newspapers, local and national news clips, social media, and academic journals. We collected 254 different food and beverage entries and broke them down into 8 categories: meat products, fresh and frozen produce, dairy, grains and beans, beverages, baking products, canned products, and snack foods. We found that meat products were by far the most affected group. Here we have a breakdown of each category and the product shortages that make them up. You can see that fresh products like chicken and poultry, milk, and fresh produce were affected the most in these categories, along with bread. Here we have the last three categories, and you can see that milk, flour, and canned vegetables are by far the most affected products for these three categories. We looked at these shortages across the United States as a whole, but also on a smaller regional level, breaking the United States into 4 regions: South, West, Midwest, and East. As you can see here, meat products were by far the highest shortage across the United States. As you can see in these graphs, the Western and Southern regions, there were no baking product shortages, while there were no beverage shortages in the Eastern regions and no dairy shortages in the Midwest regions. In all the regions, we can see a high number of meat shortages, along with grains and beans shortages in the West. We found that there were 3 main reasons behind the pandemic-related shortages: production shutdown, consumer panic, and specialized supply chain. As shown here, most shortages were due to consumer panic. In this graph, we can see that grains and beans categories were not affected by production shutdowns, while meat products were affected the most. Here we can see that although all categories were affected by consumer panic, meat products suffered the greatest shortages and snack foods experienced almost none. And lastly, we can see again that all categories were affected by specialized supply chain. However, both meat products and fresh and frozen produce were affected the greatest. Now I would like to introduce Katie, who will speak about the trends team's findings.
Hi, my name is Katie Denise, and I worked alongside Amber Ecker on the Google Trends team. So real quickly, what is Google Trends? Google Trends is a website hosted by Google that allows users to view the popularity of various search queries from Google search in either a geographic data set or a time series data set. For the purpose of this project, we specifically looked at these time series data sets. So how we developed our search queries is we aggregated the search terms from the commonly found products in the news team's search. And the list of those queries that we used, all 22 of them are on the right side of the screen. And we specifically use those terms to look into the top 34 metropolitan statistical areas across the United States in the time frame of 6 months before the 2020 COVID-19 pandemic up until October 31st, 2020. So we looked at about a year's worth of data across these search queries and these metropolitan statistical areas. And the changes of popularity of a search term could imply that there was a food shortage in a particular metropolitan statistical area. Next slide, please. So here's an example of a data set that we would pull from Google Trends. It is an example of the popularity of the query flour in the Detroit metropolitan statistical area during our specified time period. We can see that our peak is March 29th, 2020, and it's a 60% increase in popularity compared to that week prior. So it indicates that consumers in Detroit may have been worried about flour during this point in time. And we can see that the changes after this peak date are nowhere near as low as it was pre-pandemic. Next slide, please. So after we collected all of these individual search terms, we conducted cross-city comparisons among groupings of different metropolitan statistical areas. With Google Trends, we were only allowed to group 5 different MSAs at a certain point. So that's what we did here. This is an example of using the query flour against Orlando, Sacramento, Columbus, Detroit, and Dallas. In the last slide, we looked at Detroit specifically, and we saw that the peak date was March 29th, 2020. That hasn't changed in this data set, but what Google Trends does when you group different queries is it scales the data against the search query that was searched the most popularly. So here we can see that among this grouping, Sacramento, those in Sacramento searched flour more frequently than compared to the other those who searched flour in those other 4 MSAs. So we can see that those in Sacramento searched flour almost 25% more than those did in Detroit in that same time frame. These data sets also show the grouping of our peak dates. So it also helps indicate and show more information that there may have been a nationwide shortage during this time. Next slide, please. So after you did that, we also looked at the average change in search intensity 3 months post-peak. Larger numbers could indicate that there was a shortage in that MSA at a certain point. Smaller numbers could indicate that there was not a shortage or that there's still an ongoing shortage of that product. So here we've highlighted the largest changes. So in Pittsburgh and Cincinnati, searchers may have been concerned about eggs, while Sacramento, those in Sacramento were concerned about flour at some point. Next slide. Here we've highlighted the lowest points. So we see that in Portland, Las Vegas, Austin, and Cincinnati, there were there was very little change in the search intensity for milk. And for Las Vegas and Cincinnati, there was very little change in the search for flour. Next slide. Here I've highlighted just a certain term. We see that with eggs, these show the largest changes in search intensity. So it could indicate that among this grouping of MSAs, for example, that there was a nationwide shortage of eggs. Next slide. And here we see that with flour, that isn't necessarily the case. With Portland, Sacramento, and Pittsburgh, those changes were much greater than they were in Las Vegas, Austin, and Cincinnati. Next slide. Thank you. And here we're just looking specifically at Sacramento. We see that those in Sacramento, searchers in Sacramento may have been more concerned about eggs and flour than they were about milk and bread. So in closing, the data, there was a wealth of information presented here today, and the data and information presented all help to give insight into the scanner data to aid in Dr. Volpe and Dr. Tsai's research. So we thank you all for listening. Are there any questions?
Thank you. That was a great presentation, and it's very thorough. It's looking at all aspects of food pricing, food chain, looking at cities. What great work, Dr. Volpe and students, and it's awesome. So what questions? We have a few minutes here. Who has a question or comment? I mean, I have a question. Mine's a very basic question, and then I guess maybe this was for the second student that presented. Was that Camille? What is consumer panic? Does that mean that people are just buying a ton of it? Is that what that means? They're just buying it?
Yeah, so basically we define consumer panic as people going to the store panicking that food was going to run out and just buying as much as they could get their hands on.
So that was that the issue with like the toilet paper in the first beginning?
Exactly. Yeah.
And that was also for meat products too, or I was a little confused. Or is it the supply chain? There was issues with that too, both.
We found that definitely consumer panic affected meat shortages. I think meat shortages were also affected by kind of all three. So consumer panic, specialized supply chain, and then also production shutdown. So many of the meat processing facilities had to shut down early in the process due to COVID outbreaks, which then in turn slowed down the production and created a shortage of meat products that became available.
Great, thank you. Are there any other quick comments before we move on? Or questions? Thank you very much. A great presentation.
Thank you for having us.
Thank you. So as for those of you that are just joining us, welcome to the Center for Health Research COVID-19 Research Symposium. And so we're going to stick on schedule here. And so our next talk is titled COVID-19 Pandemic and Stay-at-Home Mandates Promote Weight Gain in Adults with Dr. Adam Seal.
All right, thank you, Todd. So my name is Adam Seal. I'm a postdoc. I'm actually Dr. Agobian's postdoc in the Center for Health Research, and we've worked on this project with Dr. Phelan quite a bit. And so let me share my screen, get this going. Sorry. All right, here we go. So for this study, we wanted to focus more on kind of the impact of the stay-at-home mandates that got put in place because of the pandemic, and how those stay-at-home mandates kind of affected health across the U.S. And specifically, you can see based on the title that we wanted to focus on weight gain. So just as kind of a virus timeline, just to kind of set the context of when mandates came into play and how it relates to our study. So the first US COVID case was in January 21st. And then we declared a public health emergency on February 3rd. And then the World Health Organization declared a pandemic on March 11th. And then March 19th, which is kind of where we start to think about our study, California became the first state to issue one of the stay-at-home mandates. And so just to continue that timeline out a little bit, we see California, like I said, on the 19th here. But what I wanted to show was that on around April 7th, most states, I think this is 43 here, so most of these states had implemented some sort of stay-at-home mandate by April 7th. And at this time, the US cases were just under 400,000 cases. But so focusing on our project some more. So what we wanted to, as I mentioned before, these stay-at-home mandates, we wanted to see how these changed in behavior. So what was kind of seen in previous literature is that there's been some changes in diet. So in this figure right here, we're just looking at the difference in relative frequency of use of these different products as compared quarantine versus pre-quarantine. So we can't really draw conclusions as far as weight gain with this, but I just wanted to show with this is that there are changes occurring in the diet. And then we've also seen changes as far as physical activity, and this is best kind of overall shown with this figure from Fitbit. So this week number 10 right here is the start of lockdowns. You see a 12% decrease in step count according to Fitbit, their data, their publicly available data. And that's as compared to last year at the same time. So seeing a change in diet and decrease in physical activity. So some studies before ours started to look at some of these health effects as well. Two of these, one by Flanagan, also showed increased time of sedentary and decreased activity. And then in this study as well, they showed that people with obesity had the greatest declines in mental health and the most weight gain. This was a retrospective study, meaning they took measurements, or they sent a questionnaire and then asked the participants to compare it to their past before the pandemic. And then this study by Zachary et al. Showed 22% of people had weight gain of at least 5 pounds, which we consider pretty significant, especially during this short amount of time. And they also looked at some risk factors, and those included inadequate sleep, snacking after dinner, stress eating, and reduced physical activity. And this study was cross-sectional, meaning they just looked at one point of time across all their participants. So as I just alluded to, so these studies were retrospective and cross-sectional, and neither controlled for any socio-demographic factors in their analysis. So that brings us to our study that we conducted. So our primary aim was to longitudinally look at the effect of mandates specifically on body weight in US adults over 3 months. Then we had secondary aims to explore predictors and correlates of those weight change, which included demographics, physical activity, diet, stress, depressive symptoms, sleep, and COVID perceptions, just how people perceive the virus. So getting into our methods, pretty short and simple. It was a natural longitudinal design. Any adult aged over 18 years in the US. Our survey was 98 questions, and we repeated the survey after 3 months. So just to hit on that some more, so the study timeline, first COVID cases in January, lockdowns in March. We sent our first survey in May, and then in August, we followed up with our second survey. So we're really focusing on this time between May and August. And just real quick, the statistics that we used for change in body weight, we used a paired t-test and a chi-square. And then we also ran a multivariate regression to look at the predictors of those weight gains. And we split the variables into 4 domains, which will come into play as I talk about the results some more. So just to quickly go over our demographics, we had a total sample size of 1522, pretty overwhelmingly female amount of respondents, and race was quite of 84% white, 76% Asian, 69% Black or African American, and 71% Hispanic or Latino. Household income, BMI category, and education were a little bit spread out more evenly in our population. So we kind of strive to do that a little more. We focused on recruiting to make these categories a little bit more even as far as sample size. All right, so getting into the actual results of our study. So the main figure right here, change in body weight just across all of our participants. So this is baseline. So again, that first month and then after the 3 months, we saw a 0.6 kilogram increase in body weight. And this was statistically significant between or after 3 months. And 29.7% of our participants reported any weight gain at all. And then 18.4% of participants reported weight gain greater than 2 kilograms. So then we also looked at the percentage reporting weight gain, and we split that by BMI category, just to see which BMI category they've had the most trouble. So we saw that people with obesity, a higher percentage of them reported weight gain as compared to normal weight individuals. So this kind of hints that the lockdowns could have affected people with obesity quite a bit more as far as weight gain. And these numbers right here, I know there's a lot of numbers on here. I'll kind of walk us through it a little bit. But this is just change in some of the domains of the different variables that we included. So this is just straight t-tests. So depressive symptoms increased significantly. And again, this is not related to weight gain at all. But what's interesting about these depressive symptoms is that a score of 16 is, with the instrument we use, indicative of depression. So at baseline, we were already on average indicating depression, and then that increased. Stress actually decreased, but these numbers are actually higher than the normative values for the instrument that we used. And then physical activity decreased quite a bit, about 20 minutes per day, and then sleep actually decreased, and that was significant as well. But not to, like in all real aspects, that's only about 10 to 15 minutes, so not too important there. So the 4 domains that I mentioned earlier, so demographic, psychosocial, diet, and physical activity. So this is with our regression that I'm getting into now. So we saw that only mandate length was related to weight gain. And then for psychosocial, change in depressive symptoms, then also change in time spent preparing food, and then minutes of activity per day at baseline and the change were also related. So just to interpret that, give a little more context to what all that just meant. So longer mandates were associated with weight gain, worsening depressive symptoms, more time spent preparing food at home, lower baseline physical activity, and decreased physical activity all were associated with weight gain. So our 3-month weight gain was 0.6 kilograms. Other studies have shown annual weight gain of about 0.34 kilograms to 0.8 kilograms. So that's over the course of the entire year. And then also previous studies outside of the pandemic have shown holiday weight gain of only just under 0.4 kilograms. So what we've seen, take-home message, is atypical weight gain over the 3 months of mandates. It's disproportionately impacting individuals with obesity. Longer mandates were associated with weight gain, and then physical activity and depressive symptoms were also associated with weight gain. So I just want to note this study is ongoing and we'll continue to follow participants every 3 months for the next year. So we're excited to get to analyze those data and see how the mandates continue to, if at all, improve or what's going to happen. Quick thank you to all that helped with the project, and I'm happy to take some questions.
Thank you, Dr. Seal. A great presentation. So this paper is now open for questions or comments.
Hi, Adam. I have a question. What do you think that are your predictions for the 1-year follow-up? Do you think you'll continue seeing more depression, less physical activity?
Well, I certainly hope that it gets better, some of these numbers. But I would venture to say if we continues to start opening up and everything and things start to get back a little bit more to normal, that we might see some of these numbers, especially those depressive symptoms, start to go back down to normal level. But that all depends on what happens with the virus and if anything kicks back up again. So, yeah.
Thank you.
Thank you. Other questions or comments? We still have a couple minutes.
Okay, I have another question. I know your population was mainly female and white. Do you think it would look different in other, for males or for other ethnicities?
Certainly, yes, it could. I don't want to speculate as to what it could look like, but yeah, definitely. It was a convenient sample, so it's just whoever responded, that's what we got. But yeah, it certainly could be different. Yes.
Thank you.
We have time for one more quick question. Karina, did you have another one?
I'll let other people ask. I know where I can find Adam.
Other questions? So Adam, can you just make a quick comment on the weight again, the weight gain, the 0.6 kgs and how that's, is that a lot of weight or not? Just putting it in perspective over the course of those 3 months.
Yeah, so over those 3 months, like I said, so with, like I pointed in that last slide, typical holiday weight gain is only 0.35 kilograms. So that's during Christmas and Thanksgiving. So it's kind of, we showed weight gain that was kind of double that amount. And this was just because of the lockdowns. So pretty atypical as far as that goes. And then it was also comparable to annual weight gain. So giving that context, yeah, it's pretty significant weight gain.
Great, thank you. Thank you for your presentation, Dr. Steele.
Absolutely.
Great, and so for those of you joining us, thank you and welcome to the CHR's COVID-19 Symposium. And we have our last talk in this track, in the Virus and Impact track. And the title of that is Emergency Cash Savings Accounts in a Post-COVID World by Dr. Roldson. And I'll let Dr. Roldson go ahead and start presenting. You should have access to, if you have a presentation, you should have access to that too.
Fantastic, thank you. Let me see if I can share the screen. Okay, well, great. So great to be with you. And Todd, I just want to thank you very much for giving me the opportunity to present today and share this research. It's a little bit different than some of the empirical and data-driven type of research that I know many of you engage in and present on. This will be a little bit more of a focus on policy proposals. But let's go ahead and get into it. And I would love to take any questions as we go, or if we're holding them to the end, either way is fine with me, but feel free to come in at any point. So I'm from the business school, as you see up here. Great to be with you today. I teach in the area of business law. Our department is accounting and law, but I'm in the business law area. All right, well, to start out, I thought I would ask, in 2019, for which we have data, what percentage of Americans could not afford a $400 expense? Meaning afford that expense from their savings without having to access debt or credit cards or some other source, but actually have $400 saved. I can go ahead and answer because I don't know if this is an interactive environment or not, but oh, look, there's the chat. So let me open up chat here just a minute. 30%, hey, Todd, all right, way to go. 30, 20% or so. Great guesses. I guess I need to leave chat open. Let me see if I can do this. 25, all right, Jim, right in the middle. We'll go 30, 20, 25, somewhere in there. Let me get this chat box moved over so I can see my own screen. Well, great. Well done. You guys are right in the ballpark. So 37% in 2019 could not afford a $400 expense. This is a survey done by the federal government, by the Treasury. A different survey is run with a $1,000 expense. What percentage of Americans could not afford a $1,000 expense? And we could throw numbers out as well if we would like. You know, clearly we've now doubled it from 400, even more up to 1,000. Well, 69% cannot afford the $1,000 expense. So great, I see we've got 80% and a 55% guess. So again, we've kind of got both ends of the spectrum. Nicely done. So 69% of Americans cannot afford a $1,000 expense from savings. Last quick question, and we will move forward. What percentage of Americans did not have a bank account as of 2019? Well, this one, thankfully the percentage is lower, but nonetheless 5.4% in 2019 did not have a bank account at all. That's actually an improvement. So in 2009, it was at 7.6%. So 5.4% comes out to about 7.7 million Americans without a bank account at all. So not having a bank account at all, we refer to that category as unbanked, typically in the literature on this subject. And not having a bank account certainly makes it more difficult to save. Not impossible, but more difficult. Well, a few more quick data points and questions. Total federal budget in 2019 was $4.4 trillion. We're going to be leading up to the amount of federal stimulus, which many of you may be familiar with, just because it's been in the news a lot. Well, US military expenditures in all of World War II were $5 trillion. The COVID stimulus, you know, we're probably still going to tack onto this, but we're, somewhere around 6 trillion. So these are inflation adjusted numbers, of course. It's been referred to as kind of helicopter money, which was an idea floated years ago as a stimulus measure that could be used in times of need. Well, this certainly fit the bill, and the federal government has sprung to action and provided stimulus in numerous different forms, whether it be direct payments to individuals, it's been loans on extremely favorable terms, it's been extended unemployment benefits, it's been in the form of numerous different types of stimulus, but not in savings directly, as we'll see. However, this stimulus that was provided did have an impact on savings. In fact, we had record highs in 2020. Now, if you go back and look at, other emergencies or other recessions or other times of economic upheaval, the savings rate always goes up because, people get nervous. So naturally, when you don't know if you're going to have a job next month, this this month you're going to save, you're going to hold on to your funds. So it's no surprise that it would go up, but it went up much higher than a typical emergency situation, in large part due to the stimulus. So that's great news. However, looking more closely at the savings, one realizes that the savings are largely wealthier people saving more money. And so we did see in March and April, the bottom 40% of earners actually increased their share of total cumulative savings. So, for a brief moment, the savings blipped up for lower income people. But since then, the savings of the bottom 40% are depleting more rapidly. Not terribly surprising, just in the sense that wealthier people are going to be able to cover their expenses, their daily expenses, more easily and have money left over. So not terribly surprising that wealthier people would save more. However, from a policy perspective, it's important to ask the question how or whether the stimulus funds that we provide target the right goals or not. And at the time of the initial stimulus coming out, a lot of the proposals were talking about the importance of stimulating the economy. And there was even a proposal floated about instead of sending cash, which can be saved, maybe the government should send a use it or lose it debit card to basically force people to use the stimulus funds to spend the money and thereby stimulate the economy. Go to restaurants, buy things at the store, which all helps, with jobs and things. So there's good reason why stimulus funds are designed to be spent. However, the question that I really investigate in this article is it really the case that we should have no provision for future savings? Or said it differently, is it possible that maybe the COVID emergency could be a lead-in to preparing better for future emergencies through increased savings rates? So let's look a little deeper at savings. And I just want to look at my time here. Todd, I want to stay on track. So got 5 minutes or so, is that about right?
Yeah, so you want to leave a few minutes for questions. So you have like maybe 3 minutes. Can you?
Got it.
Yeah.
Let me zip us through. So why not save in a checking account? Well, we refer to a double tax on savings, where you are taxed on the money you earn. So if you earned $10 and paid a 20% tax, you'd have $8 left over to invest. When you invest that, you then pay tax on your earnings. So that's the double tax that we refer to. So that's the first reason why it's less efficient to save in a standard checking account. But then secondly, it's important to note that we do have, tax favored savings accounts, and, IRAs and 401ks are things that many of us would be familiar with. There's numerous other types of savings accounts. Well, another reason why these are important, apart from eliminating the double taxation issue, is that people save more when there is a defined pathway to do so. So I posit in this article that emergencies are also foreseeable, not just retirement, college education, medical expenses. Yes, they're foreseeable, but emergencies are too. They strike indiscriminately. Their timing is unpredictable, but they do come typically to us all. So the proposal would be to have, similar to an IRA or a 401k, but a much smaller dollar amount where people could save a small amount per year to prepare for future emergencies. And I will skip over some of this to go quickly. Some of the details of the proposal would include a matching tax credit, where currently what exists is a saver's tax credit, where a person can get a tax refund for taxes paid. What I would propose is a matching tax credit that would be paid, that would be deposited directly into the electronic savings account, so that the tax credit is not just sent back as cash that would then be spent, but rather is deposited into the account. In addition, one of the big issues with designing savings accounts is access for unbanked people or low-income people who often find that banks create obstacles for them to open accounts, such as minimum balances, such as fees. And because of free electronic accounts that are essentially open to anyone, regardless of race or credit history or background, and they can be opened online, it's now feasible to create free accounts that could be used for this purpose. There would be a few other details here, but I'm skipping over them a little bit quickly here, just so that we can get to the kind of final proposal in a nutshell. You know, can COVID be a springboard for Americans to save better, to prepare for future emergencies? Well, these 5 characteristics, I think, would help us get there. Make it a relatively low amount, give tax savings for emergencies. Raiding an IRA or a 401k before retirement to cover emergencies is not a very good method for addressing emergency expenses. So in addition, an emergency account could provide much more easy access to the funds, whereas for obtaining access to 401k funds for emergencies has many more hurdles that are necessary. So having an account designed for emergencies would allow for much quicker access. I'm going to go ahead and leave it at that. This is very much just a nutshell of the proposal, but hopefully you see the concept, and I would love to discuss further or address any questions or comments that you might have.
Thank you very much. That was great. Very provocative. Are there any questions or comments? So I actually had a question, and so so this is definitely outside of my area of expertise, but if individuals received a stimulus, and let's say they bought stocks on the open market, where does that fit? Is that more of a savings, or is that an expense? Like, how does that, or if they got, let's say, the debit card, you mentioned a debit card, can you buy stocks with that? And how does that stimulate the economy too by buying stocks? It's a loaded question.
Yeah, great point. I mean, I think the short response would be that I don't think it provides much of a stimulus effect, but it would count as savings. So if a person invests in stocks, that would be recorded as saving the money. And whether that could be accomplished with a debit card, it would probably depend on the way the debit card was structured. But typically when people make the debit card proposal, they're doing it to prevent people from buying stocks with the money and instead get them to use it at Walmart, or Home Depot or whatever.
And I don't know what the investment rate, like I don't follow stocks that well with stocks, pre and before the pandemic or when it hit, but it seemed like more people were interested in the stock market once the pandemic hit and the prices went down. My guess is that a lot of people invested in that, and maybe the data on that.
Yeah, great point. I mean, I won't be able to whip out figures on that, but I can definitely affirm that it would have been a great time to invest. I mean, people who were able to take the stimulus funds and invest it immediately, it was great timing simply because we had pretty much a trough, and so that would have been a great move. The intent of the stimulus was more related to stimulating spending as opposed to saving, which was appropriate at the time. Like, I'm not saying that we should have been focused on saving at that moment. However, seeing what we went through with the emergency stimulus, it seems that it's an opportunity to look towards mechanisms to kind of grease the skids for savings, make it easier for people to prepare for emergencies in the future.
Well, thank you very much. That was a great presentation and very insightful. We appreciate that. And so thank you. And I want to thank
you
everyone.
so much, Todd. I appreciate it.
Yeah, and I want to thank everyone for attending the CHR COVID-19 symposium. And again, this will be recorded and posted on our website online along with the abstracts. And with that, we're going to end our session today, unless there's any other questions or comments. Great. Well, thank you very much. I appreciate it all, and good luck in all your future work.
Thank you, everybody.
Thank you, everyone.